Good morning, everyone, and welcome to today's event of the American Physiological Society entitled An In-Depth View of Health Disparity Through Chronic Disease Research. I will be serving as your moderator today. My name is Dr. Eric Bolland-de-Chantemelle, and I am a member of the American Physiological Society and a professor of the Vascular Biology Center at the Medical College of Georgia. In today's session, our speaker, Dr. Moira Bouchard, Dr. Kwabi Addo, and myself will discuss impactful research currently being conducted in the area of HIV, cardiovascular disease, and cancer within the context of underserved and vulnerable population. A few housekeeping notes. Out of respect of our speaker, we ask that you refrain from taking photo or video capture of the presentation in whole or in part. This session will be recorded and available for future viewing through a link on the registration website. Recording or taking photograph of another person's presentation without their explicit permission is prohibited. At this time, all attendees, except for the speaker, are in listen-only mode with video off. As moderator, I will be happy to raise questions that would have been entered into the Q&A function on the bottom of your screen. You may put your question in the Q&A at any time. The session will end at approximately 12:30 p.m. Eastern Time. It is now my pleasure to introduce our first speaker, Dr. Jessie Moira Bouchard. Dr. Moira Bouchard is a co-PI of the Boston University Queer Understanding, Equity, and Education Research Lab. Their research and teaching focus on LGBTQIA2S+ equity and inclusion in STEM and physiology education, as well as cardiovascular health risk in LGBTQIA2+ community. Dr. Moira Bouchard, welcome, and the floor is yours.
It's wonderful to have this opportunity to share with you all my research today, in part, and additionally to contextualize an emerging problem through some other areas of research in the field of minority health. So let me just share here. I'd just like to thank the APS for hosting me as always. It's wonderful to get to work with this lovely organization who I think care a lot about health in general and in marginalized communities. So today I'm going to talk to you about enabling access to exercise as a therapeutic for the emerging physiology of psychosocial stress-dependent hypertension. So in brief, we're going to go through an introduction to hypertension. And then we'll get to CVD risk in LGBTQIA+ communities. And finally, we'll end at enabling exercise as a potential medicine here. So to begin, and just kind of give everyone a nice overview, hypertension is an extremely prevalent disorder that's estimated to affect 1 in 2 adults in the United States. And notably, one-fifth of people are treated with lifestyle modifications, and 4/5 of people with hypertension additionally have a medication therapy, but 3/4 of people still remain uncontrolled with respect to their blood pressure. So hypertension is not only a major prevalent disorder, but one that's currently poorly managed. And as we start to break it down by populations, you can start to see that there are actually differences across subpopulations, and I'll present some of those data today, where there are even worse risks than just the general myocardial infarction risk, chronic kidney disease risk, based on that subpopulation related to identity. Now, additional background that's relevant for today is the minority stress theory. Elon Meyer published this revolutionary meta-analysis in 2003 that really pulled on some of the recent pieces of psychosocial stress literature and demonstrated that lesbian, gay, and bisexual people have a higher rate of mental disorders such as anxiety and major depressive disorder. And it posited that stigma, prejudice, and discrimination create hostile or stressful social environments for these folks that is actually the root cause of these mental health problems. And this was revolutionary, even though it was just in the LGBT cisgender subpopulation, because it wasn't really something that people were focusing on until this time in any meaningful or large way, meaning, how do minorities, sexual and gender minorities, experience cardiovascular disease, let alone mental health disorders. And this has been extended since 2003 to multiple different LGBTQ populations. Here I have a figure from Dr. Billy Caceres' Circulation 2020 AHA scientific statement, where they posit that not only does minority identity, particularly being LGBTQ, associate with these various psychosocial factors like depression, stress, anxiety, but that additional behavioral factors that may be coping mechanisms like increased rates of tobacco use, poor diet quality, and then related factors like being sedentary, having a low physical activity score, underlie a progression beyond just these mental health problems to physical health problems like obesity, hypertension, diabetes, and dyslipidemia, high cholesterol, ultimately driving enhanced rates of cardiovascular morbidity and mortality. And so this theory is continuing to be built between having a minoritized identity, experiencing adverse interpersonal and structural stressors, developing mental health problems, and then ultimately developing physical health problems. But what actually are the data that support this? So that's where we kind of move to what is our current understanding of cardiovascular disease risk in these queer communities. So when we first think about hypertension, we have some data from a paper, Billy Caceres group in 2022. And that's first that gay men were 18% more likely to have been diagnosed with hypertension. Additionally, bisexual women were 19% more likely to have been diagnosed with hypertension. And notably, bisexual women were 29% less likely to be on antihypertensive medication therapy. These are all with respect to same gender heterosexual peers. And so there is an increased prevalence of hypertension in multiple populations within the LGBTQIA+ community. When we look at the recent Life Essential 8 score, so this is the American Heart Association's kind of cardiovascular health level score, right? It's a composite of several different cardiovascular related variables. We can see that at this composite level, bisexual women and lesbian women and women who declined to answer related to their sexual orientation had worse CVH scores than their heterosexual peers. Although interestingly, gay men and bisexual men had better CVH scores than their heterosexual peers. And so there's kind of this notion where multiple different papers have different indices related to cardiovascular health, and you end up with different findings. When we look at one more piece of critical data, we can see that from this 2019 study, both transgender men and transgender women have worse adjusted odds ratios of myocardial infarction compared to cisgender folks, suggesting that not only are hypertension and cardiovascular risk elevated in queer communities, but adverse outcomes like MI, which of course can progress to situations like heart failure, are also increasingly appearing in these minoritized populations. But a lot of these studies, when we really reflect on them, are limited by the fact that they are retrospective in nature and look at electronic health records. They may be national surveys, but these surveys weren't designed with the appropriate sexual orientation and gender identity identifiers. And so there's kind of an incomplete ability to parse out people into true subcategories based on their identity. Instead, a lot of what we call lumping happens. And so we can only say something about a broad community of multiple different subpopulations. And in addition, a lot of the data collected in surveys are self-report health data. So questions like, has a physician or other healthcare provider ever told you you have high blood pressure, also known as hypertension? And these questions can be worded in more reliable ways sometimes, but they're not as great as direct data acquisition in living, breathing people who come in for a study visit in front of you. Now, before I kind of get to where we think we might want to head, there's also a problem of exercise, right? We all think, oh yeah, cardiovascular disease. Well, are these populations active? You mentioned physical sedentary behavior a couple slides ago. Well, there are some, positive effects of exercise on cardiovascular disease. We know that cardiovascular disease and even psychological stress are reduced by exercise. There's normalization of autonomic control, so rebalancing of parasympathetic and sympathetic control of cardiovascular function, regulation of the baroreflex. There's glycemic control that's achieved through regular exercise and regulation of the daily rhythmicity of cortisol release. But why are these populations sedentary? And is that related to their outcomes? There was a report from the LGBT Foundation in 2016, which was nearing 10 years old, but not much data has appeared since then on these measures, suggesting that 52% of LGBT people do not meet government recommendations for physical activity in a given week. And additionally, about 17% of survey respondents in this study were members of a sports club or team, with 48% of these being members of LGBT-specific clubs, and 3/4 of the queer people who were members of such sport clubs met the national recommendations, compared to 38% of queer folks who were not members of sports clubs. So while there is probably a benefit of having cardiovascular exercise, these populations aren't very likely to be participating at physical activity levels needed to improve their health, unless the few of them are a part of these kinds of community group-based organizations, which probably provide some sense of belonging for them beyond additional use of standard gym facilities. So why are very few of them accessing these kinds of spaces, and what are the barriers? This is something that my co-PI and I in our lab think about a lot, the cardiovascular health of LGBTQ people and other minoritized groups. And in particular, the biomedical aim of the two aims of our lab, the other being educational, has a focus in the third point there. What are the mechanisms of psychosocial stress on cardiovascular health? And kind of the extension of that is, what are the ways that we can enable access to things that are already understood to ameliorate cardiovascular problems? And so again, in the context of this notion that stressors drive adverse mental health effects and adverse coping mechanisms that create disease states, how can we kind of block this? Well, we hypothesize two things. The first being that queer identity is associated with higher blood pressure beginning at a younger age as a function of increased stress, anxiety, and depression, and that exercise inaccessibility is probably associated with higher stress and higher blood pressure in queer young adults. Focusing, again on young adults, because while the general population is estimated to develop hypertension, if you're experiencing this stress for your whole life, it's quite likely that you'll develop hypertension and the subsequent adverse cardiovascular outcomes earlier. And so, having just started our lab this year, we don't quite have data yet, but I thought I would share our approach with you. First, honing in on straight, lesbian, gay, or bisexual, cisgender men and women between the ages of 18 to 35, so young adults. Want to bring them in for study visits and even just assess first blood pressure levels, demographics, comprehensive health history, physical activity questionnaires, additional blood pressure measurements, as well as depression, anxiety, and stress survey scores to get a kind of cross-sectional association of blood pressure levels with mental health, and then even take some waist and hip measurements and see basically what are the associations that exist in these populations between blood pressure levels, stress levels, and perceptions of physical activity, attitudes towards physical activity, barriers to physical activity, if there are any, because we hypothesize that there's going to be an association, again, of lower physical activity levels with worse mental health outcomes, which associate with worse blood pressure levels. And, we think that there will be some interesting data to find here that hopefully will point us in a direction towards enabling exercise as a potential beneficial therapy in addition to medication therapy for folks who have psychosocial stress-related hypertension. So, what are the takeaways in thinking about the problem, really, right? Because there aren't solutions yet. They're that queer communities are at elevated risk for developing cardiovascular disease and having adverse cardiovascular disease related outcomes. This is true and demonstrated, although we need better study designs that will allow us to get longitudinal understandings of these phenomena in addition to cross-sectional or retrospective. Probably that culture and social change is needed to address the root of the elevated disease risk, because while we can find good therapies that will ameliorate autonomic dysfunction or cortisol elevations, the root of the issue is that there are psychosocial stressors driving the disease state, unlike the general essential hypertension population who may have some unidentified genetic risk factor. And that the LGBT Foundation study points to an effect of community level programming that fosters sense of belonging to enable physical activity to occur in LGBTQ plus populations. So we kind of have to have a structural approach and a grassroots level approach to work with this population in the hopes that we can address a critical and unmet issue here. All right. With that, I hope I've left you with something to think about. And again, thank you for having me today. And I just want to thank my co-PI, Dr. Lisa Roberts, our lab student, Evan Nesson, and all the folks who we work with on a day-to-day, and my department for really enabling us to ask these critical questions for today's contemporary and burgeoning emerging physiologies. Thank you.
Well, thank you very much, Jesse, for that wonderful presentation. And now we will move to our second speaker. Then I would like to introduce Dr. Bernard Kwabi-Addo. Dr. Kwabi-Addo is a biochemist and molecular biologist who has worked in the field of cancer research, in particular prostate cancer research, for 20 years. Most of his training and research work has focused on elucidating the genetic and epigenetic alteration underlying cancers and the disease disparity. Dr. Kwabi-Addo, we look forward to your talk.
Thank you very much, Eric, for the kind introduction. And I want to thank the APS for the opportunity to come and present my work, looking at the role of epigenetic alterations in prostate cancer disparities. So I want to begin by presenting this slide that shows the mortality rate associated with diseases in America. So the top 10 leading causes of mortality are presented here. This is heart disease, followed by cancer, unintentional injuries, cerebrovascular disease, Alzheimer's, diabetes, kidney disease, and suicide, making the top 10. However, when you stratify the disease incidence and mortality rates by race and ethnic groups, we see that African American are disproportionately impacted by many of these diseases, such as heart disease, cancer, cerebrovascular disease, diabetes, pneumonia, and influenza, and kidney disease. Of course, not all the diseases disproportionately impact African American or US Blacks, as shown by the higher incidence of chronic lower respiratory disease in European American individuals compared to African American individuals. However, in terms of the health outcomes for the average US Black, the overriding theme is that of disparity and disadvantage. As a result, the life expectancy for US Blacks has persistently lagged behind that of their Caucasian counterpart for 100 years, if not more. And I guess the recent disparities that we saw with regards to COVID-19 pandemic is a clear indication of the persistent challenges that we face with regards to health disparities in the US. And I appreciate the presentation from Jesse that talked about the disproportionate disease burden in the LGBT plus community. So in health outcomes in a foreign land, I actually discuss the social determinants of health, the genetic and the epigenetic interactions, as well as the environmental exposures that is driving some of these health disparities that we see in our US population. But for today, I'm going to focus my work, my talk on prostate cancer, and I'm trying to advance this, if I can advance my slide. Bear with me for a second. I don't know what's happening. I can't advance my slide. Okay, let me.
Dr. Quabiado, you may need to click one time on your slide.
I think so. That's what I'm doing right now. Yes. Okay. So I'm going to focus on the disparities associated with prostate cancer disease. So prostate cancer continues to be a major public health. This year is estimated that more than 288,000 men will be diagnosed with the disease. And even though it's considered to be a slow-growing disease, it's also estimated that about more than 34,000 men will die from prostate cancer in 2023. Some of the well-established risk factors for prostate cancer are age, as most men diagnosed with prostate cancer over the age of 50. Individuals of African ancestry are disproportionately impacted by prostate cancer. The incidence rate is about twofold higher than their Caucasian counterpart, and they die from prostate cancer at about 1.6-fold higher than their Caucasian counterpart, who ranks second in terms of prostate cancer incidence and mortality rate when you stratify by race and ethnicity. Having a father, maternal uncle, or siblings who have prostate cancer increases your risk of the disease, suggesting a very important familial predisposition component to the disease. And there are other factors such as socioeconomic status, certain environmental exposures, lifestyle risk factors, including smoking, excessive alcohol binging, which contribute to prostate cancer. And also access to healthcare plays a very important role in the disease disparity. Some studies report that if equal access to healthcare is provided for all groups, this significantly reduces the disparity that is seen with prostate cancer. But in addition to all these factors, we know that biology, particularly genetic and epigenetic alterations, is also a very important factor for prostate cancer. Yeah. So recent large-scale genome-wide studies, integrative genomic analysis is really helping us to be able to identify the prevailing genomic alterations that are associated with the disease. The challenge of prostate cancer is that it's a very heterogeneous disease, and there are not well-identified predominant signaling pathways, and this poses a challenge with regards to coming up with efficacious treatment for different ethnic and racial groups. But some of the top leading causes of mutations that are identified includes the temperance egg fusion. This is seen about 50% of prostate cancer from individuals of the Caucasian or European American race. And this temperance egg fusion is very low in US Black men. On the other hand, US Blacks have higher frequencies of mutation FOXA1, SPINK1, P53, and the proto-oncogene MYC, driving it to the oncogenic variant. In addition to the genetic alterations, there's a lot of epigenetic changes, including mutational changes in several genes that are expressed differentially in Blacks versus whites. This includes the PMEP-A1, retinoic acid receptor 1, beta promoter, and SNRPN, which plays a role in RNA splicing. So these are some of the genes that we have reported to show differential methylation in prostate cancer from African American compared to European American. And these have significant biological consequences, as I'll talk to later on. So again, the knowledge about the genomic variants is helping us to really be able to come up with efficacious therapeutic intervention. And some of these variants allows us to really come up with subtypes of prostate cancer. So typically we have the ETS positive prostate cancer. This is typically seen in individuals belonging to European American population, and the so-called ETS negative, where we begin to see additional mutations such as SPOC and FOXO1 and other genes. And most of this early localized subtype prostate cancer respond very well to androgen deprivation therapy, which still is the mainstay for treating prostate cancer. Unfortunately, a subgroup of this localized prostate cancer will become resistant to the androgen deprivation therapy, and will obviously progress into metastatic prostate cancer, known as the castration-resistant prostate cancer, when they no longer respond to androgen deprivation therapy. And with the transition from the localized prostate cancer to metastatic prostate cancer is actually associated with additional mutational changes, including androgen receptor gene amplification, splice variants, and other genomic changes. So this, of course, plays a very important role in coming up with additional therapy that would be able to treat individuals with the castration-resistant prostate cancer. In addition to that, studies have shown that certain mutation plays a very important role in being able to determine the survival probability of individuals that have undergone some sort of some surgery. And here we see one study showed that there's a significant role of PTEN mutation that is linked to increased risk of lethal prostate cancer. However, this risk of lethal prostate cancer in individuals with PTEN mutation is only significant when we also have egg negative mutations. So a lot of these changes at the mutational level is really providing information with regards to men who have undergone surgery or some sort of treatment and how their survival pattern or probability will be reflected as a result of some of these mutational changes. But in addition to the genetic changes, we also know that epigenetic alterations, and I should say abnormal genetic changes, significantly influence prostate cancer risk and the disease progression. As we all know, genetic changes plays a very important role, particularly during development, in establishing cell type diversity and also promoting heritable cell type identity. Using phenomenon known as DNA methylation, histone methylation or acetylation, and also non-coding RNA. However, increasingly, we see that a lot of abnormal genetic changes actually is seminal to the early stages of prostate cancer and giving rise to genomic instability and, of course, causing a lot of additional genetic alterations that is driving the disease progression. So why study epigenetic changes? This is the work that my lab is focusing on. So in 1995, David Barker hypothesized the fetal origins of adult diseases, noting that what the growing fetus get exposed to in utero has profound effect on the growing, not only on the growing fetus, but throughout the lifespan of that individual. And this effect can actually be transgenerational, up to 3 or 4 generations. So what we get exposed to with regards to diet, certain lifestyle, if a pregnant mother or the father is a smoker, or exposure to alcohol, some sort of environmental pollutants, hormonal exposures, these are all the factors that can contribute to changes in the of the growing fetus using epigenetic phenomenon. And there are various studies to support some of these epigenetic changes, as we see from genome-wide methylation studies in cord blood from newly born black versus white babies. They show significantly differences in the epigenetic pattern or DNA methylation pattern. Similarly, there are a lot of differences in the epigenetic regulators that have been reported in cancer. And also we see distinct tumor microenvironment changes in prostate cancer from African-American compared to their Caucasian counterpart. So the generational effect of some of these genetic changes has been reported with the recent Rwanda Tutsi genocide, where specific methylation regions have been shown to be associated with mental disorders like PTSD in not only the mother, but 2 and 3 generations subsequently. So we see that epigenetic plays a very, very important role in the disease etiology as well as progression. Some of the early work that we've done in the lab is to actually look at DNA methylation in normal prostate tissues. And here we show that DNA methylation actually increases as a function of age in a lot of in organ donor samples from these are normal prostate tissues from men, suggesting that this increased risk of or increased methylation pattern could be a precursor predisposing to prostate cancer risk. And nowadays, DNA methylation is actually used as a biological clock to really determine the rate of aging as a result of various environmental exposures. So again, DNA methylation is playing a very, very important role to help us to assess some of the biological determinants that are adversely or positively influencing how we age, including diet, BMI, exposure to all sort of environmental pressures. Again, some of the early studies that we've done looking at the role of genetic changes in prostate cancer have actually focused on using radical prostatectomy samples where we analyze loci-specific methylation changes. And here we looked at 6 genes, GST pi androgen receptor, retinoic acid receptor, and other genes. These are potentially putative tumor suppressor genes. And we see significantly higher methylation or hypermethylation in the cancer versus the normal adjacent tissues. And overall, we see that a lot of this methylation pattern was significantly higher in the African-American samples compared to the European-American samples. Now, this phenomenon is not unique to prostate cancer, as we observe similar differences in the methylation pattern, whereby we identify cadherin 13 to show significantly higher methylation in samples from African-American compared to the European counterpart. And as I mentioned earlier, this methylation changes has important biological consequences. So here we looked at the biology of 2 genes, ABCG5, a transporter gene, and again, as I mentioned, SNRPN, we plays a role in RNA splicing. And we have reported differential methylation of these 2 genes in prostate cancers from African-American versus Caucasian-American. So when we knocked out these genes in a panel of cell lines, PC3 and LINCAP cells, these are cancer cell lines derived from white individuals, and MDA-PC is a cell line derived from African-American individual. Knockdown of ABCG and also CNRPN show significant inhibition of cell proliferation as well as cell invasion in the cell lines from the Caucasian counterpart. We do not see this profound effect in the cell lines derived from African-American, suggesting that as the oncogenic effect of SNP, SNRPN, and ABCG5 has more profound effect in prostate cancers from Caucasians, whereas we don't see such profound effect in prostate cancer derived from African-Americans. So clearly, methylation is influencing the biology of several genes to modulate the milieu of or the microenvironment of prostate tissues. And we believe that some of the significantly higher or aberrant methylation that is seen in African-American compared to Caucasians could potentially be driving the aggressive prostate cancer that is seen in the African-American population. So more recently, we have done more genome-wide methylation studies, and in addition to transcriptomic analysis. And this allows us to really compare our data with the existing TCGA, which stands for the Cancer Genome Atlas. This is a comprehensive studies that have both the methylation, protein data, transcriptomic data, and so many other variables. However, a lot of the TCGA data sets doesn't have a lot of African-American samples in the studies. So we are fortunate to be funded by NIMHD to actually try to bridge some of those studies. So here we carried out our methylation analysis in transcriptomic and we're able to compare it with the existing large data sets such as the TCGA data sets. First of all, we look at the methylation profile in the genomic architecture from blacks versus white samples. And we see that in Blacks, a lot of the methylation occurs in the intergenic or non-coding region of the genome, whereas lower percentage is seen in that region. On the other hand, in the TCG data sets, which is from the European American cohort, we see a lot of the methylation profile occurs in the transcriptional start site or regulatory regions, more so than seen in the African American samples. And also the analysis enabling us to be able to see if we can identify certain genes that are preferentially methylated and be able to address differences or as a ranking for Gleason score or tumor grade. So we identified about 20 such genes that are able to differentially distinguish between normal prostate tissues and different tumor grades. So this clearly is a marker that can be explored for prostate cancer detection. And pathway analysis showed 2 key signaling pathways that are altered based on our genomic studies, rather. So tumor microenvironment pathway, this really is consistent with what has been reported previously. In addition, we see that this methylation affects glucocorticoid receptor signaling. And we all know that glucocorticoid signaling actually plays a role in the secretion of cortisol, which is the stress hormone. So clearly, methylation is a lot of this environmental stresses is using or inducing aberrant methylation using this glucocorticoid signaling pathway to obviously exacerbate prostate cancer risk in the African American population. So we are increasing our analysis, more genome-wide studies, and here we have different prostate samples, and also be able to investigate if we can potentially study a lot of this methylation as potential diagnostic marker for prostate cancer. So here we see that they cluster very well, giving us the hope that we can potentially investigate all these methylation changes as potential markers for disease detection. And so talking about disease detection, some of the work in our lab is obviously exploring some of these markers in the cell-free circulating tumor cells from African American patients. Here we show that certain genes, including retinoic acid receptor SPARC, can potentially or significantly associated with increased PSA, which is a marker for prostate cancer, and also BPH, I should add. And also some of these genes significantly are linked to Gleason score. Again, we show that methylation changes can be explored for aerobic exercise events, and not only in tissues, but also in different biospecimen. Here we show that we can investigate DNA methylation marker in saliva-based samples, and increasingly we are interested in looking at DNA methylation changes in blood, urine, and here I showed in saliva samples. So I think, of course, methylation plays a very, very important role in the disease progression, and I think that's going to help us to really explore that as a tool, not only for detecting prostate cancer in tissues, but more so in non-invasive prostate cancer tissues. So I'm going to stop here, and this is my acknowledging slide, and thank you for your audience. Thank you.
Thank you very much, Dr. Kwabiado, for this wonderful presentation. To our audience member, please note that questions for our speaker will be answered during the upcoming Q&A session. I will now begin my own session. I'm Eric Blanchotmel. I'm a vascular biologist by training, and my research over the last 15 years has focused on the understanding of the mechanism of hypertension in understudied population, including obese women and people with HIV. And this is on this HIV population that I will now focus in the next 20 minutes. Let me share my slides. And the title of my talk is T lymphocyte-derived HIV protein induced endothelial dysfunction and hypertension in a mouse model of HIV. Then the question that you may have for me is why focusing on HIV when in the last 3 years, our whole world has been severely impacted by the COVID-19 epidemic, and most of the vulnerable population have been, in fact, severely impacted. The answer to this question is, in fact, quite simple. While HIV is not is not on the headline of all the newspaper as it had 40 years ago, HIV is still a worldwide epidemic. If we look at epidemiological data, we can see that currently 38.4 million people live with HIV. And these 38.4 million people, it's the size of the population of a country like Canada, Morocco, or Saudi Arabia. And what is very concerning about those numbers is the fact that despite all the knowledge gained on HIV in the last 40 years, and despite all the measures developed to prevent the transmission of the virus, the number of people living with HIV is still increasing. If we look at the epidemiological data here in the United States, we can see that 1.74 million people live with HIV. And we can also see that the number of new cases of HIV is around 67,000. The alarming aspect of those data is the fact that most of the individuals getting infected with HIV are among vulnerable population. If we look at those data provided by the CDC, we can see that 75% of those new cases of infection are among minorities and people of color. We can also see that more than 40% of new infection in the United States occur among Black people. If we dig more into the composition of those population, we can see that new HIV infection disproportionately affect gay and bisexual men and Black heterosexual women. On a more positive note, then thanks to the advent of combination antiretroviral therapy, the life expectancy of people living with HIV has markedly increased. We can observe in this graphic that in the span of 10 years between basically the year 2000 and 2010, the life expectancy of people living with HIV has increased by almost 20 years. With this increase in life expectancy, we have seen a shift in the cause of mortality in this population. Patients with or people with HIV are now dying from cardiovascular disease when they used to die from opportunistic disease. And cardiovascular disease is now the leading cause of deaths in people living with HIV. And high blood pressure or hypertension, which is the leading cause of cardiovascular disease, affect almost 68% of people living with HIV. And again, if we look at epidemiological data, we can see that the prevalence of patients with HIV with hypertension is higher among minorities and African American. And similarly, we can see that the control or that blood pressure control in African American is lower than in the white population with HIV. And those high rates of hypertension and those lower rate of blood pressure control leads to increased number of cases of hospitalization for cardiovascular disease. And this is clearly illustrated on this graphic showing that African American with HIV and AIDS have much higher incidence for hospitalization for cardiovascular disease compared to white individual with HIV. Then also we know that cardiovascular disease is a primary cause of death in people with HIV. The mechanism whereby people living with HIV develop hypertension remain unknown. However, two theories have been proposed. The first theory is that antiretroviral agents themselves exert side effects that can lead to an alteration of the vasculature and promote the development of cardiovascular disease. The other theory is that the HIV virus that is still present, although repressed, is releasing viral protein that can alter the vascular system and the cardiovascular system in general, and then promote the development of cardiovascular disease. And this is the second theory that we wanted to test. And the major rationale for us to test this hypothesis is the observation that elite controllers have an increased prevalence of cardiovascular disease. Elite controllers are a subpopulation of people living with HIV who are able to repress the virus on their own. They do not need to have antiretroviral therapy. And despite the fact that they are able to repress the virus, that they are not on antiretroviral agent, they have an increased prevalence of cardiovascular disease. Then based on this evidence, we propose a hypothesis that T-cell-derived HIV protein contribute to HIV-associated hypertension and endothelial dysfunction. To test this hypothesis, Taylor Kress, who is a PhD candidate in my laboratory, designed a protocol involving the use of a transgenic mouse model, the TG26 mouse. The TG26 mouse ubiquitously expressed 7 out of the 9 HIV proteins. The major advantage of these mice is that they don't express GAG and POL, which makes the virus infectious. And then we have a mouse model that is safe to use in the laboratory. One of the caveats of this mouse model is that those HIV proteins are not restricted, or their expression is not restricted to immune cells. And as you can observe on this graphic, here we measure the expression of the TAT protein in different tissue in both male and female mice. And we can see that those viral proteins can be expressed in tissues such as the kidneys, the liver, the aorta. Then to try to circumvent this limitation, Taylor designed a protocol involving the use of either intact TG26 mice and their legitimate control, or the use of animals that have been submitted to bone marrow transplant to have the expression of those viral proteins only in hematopoietic cells. Then he transplanted bone marrow from TG26 mice into wild-type mice, but he also used mice in which bone marrow has been transplanted from wild-type mice into wild-type mice as a control group. And he also generated a rescue experiment by transplanting wild-type bone marrow into TG26 mice. Then the animals of all those groups were used to measure blood pressure. Then blood pressure was measured via radiotelemetry when we use intact animals, and we use TELCOF for the animals submitted to bone marrow transplant. At the end of the blood pressure recording experiment, then we isolated the vessel of those animals, and we mostly focused on the mesenteric artery, which are resistance artery involved in blood pressure regulation. Then the first experiment that he did consisted in measuring the blood pressure in the intact TG26 mice and their legitimate wild-type control, and he used both male and female animals. And as you can appreciate on this graphic, he observed that the expression of viral protein, or the ubiquitous expression of viral protein in both male and female mice, leads to an elevation in blood pressure, suggesting that viral protein could potentially elevate blood pressure. To determine whether this elevation in blood pressure is mediated by viral protein expressed in hematopoietic cells, then he measured blood pressure in our animals that have been submitted to bone marrow transplant. And as you can appreciate, when he transplanted bone marrow from TG26 mice into the wild-type mice, he observed an elevation in blood pressure that was also observed in the female mice. When he did the rescue experiments by transplanted bone marrow from wild-type animals into TG26 mice, he observed a reduction in blood pressure, and this was observed both in the male and the female animals, suggesting what those two experiments basically demonstrate that expression of viral protein in hematopoietic cells leads to an elevation in blood pressure. We were then interested in investigating what could be the mechanism leading to this elevation in blood pressure. Since we are a vascular lab, then we focused on the vasculature. We isolated the mesenteric artery of those animals, mounted them on a wire myograph, and looked at the relaxation induced by acetylcholine. And as you can appreciate, the vessel from the intact TG26 mice in yellow, either male or female, present an impaired relaxation to acetylcholine, suggesting an impaired endothelial function. To confirm that the impairment came from the endothelium, we repeated the experiment with sodium nitroprusside that induced the relaxation of the smooth muscle cells. And as you can appreciate, sodium nitroprusside response was not different between wild type and TG26 mice, indicating that the dysfunction is at the level of the endothelium. We repeated the same experiment with the vessel from the animals that have been submitted to bone marrow transplant. And in red, we have the animals receiving the bone marrow from the TG26 mice. And as you can appreciate, expression of viral protein from hematopoietic cells leads to an impaired relaxation in both male and female, and then doing the rescue experiment by transferring bone marrow from wild-type mice into TG26 mice restore the relaxation, suggesting that viral protein expressed in hematopoietic cells are the cause of this impaired endothelial function. Then from there, we propose that hematopoietic cells expressing HIV protein leads to endothelial dysfunction and hypertension. The following question that we wanted to address is which hematopoietic cells contributes to this elevation in blood pressure and this endothelial dysfunction. For that, Taylor developed a new model consisting in collecting T cells from the spleen of either TG26 mice or wild-type mice, putting those T cells in culture for several days, and then exposing the aorta of wild-type mice to those T cells. Then when he did this experiment, and then look at the consequences of this exposure to T cells on endothelium-dependent relaxation. And as you can appreciate, when he exposed those wild-type aorta to TG26 T cells, he induced an impaired relaxation, suggesting that T cells expressing viral protein induce endothelial dysfunction. To test the functional relevance of those observations, he went back to the intact TG26 mice, instrumented them with a telemeter, and measured blood pressure in response to abatacept, which is a CTLA-4 inhibitor and inhibits T cell activation. When he did this experiment, he induced a reduction in blood pressure in TG26 mice in response to T cell inhibition. Similarly, what he observed here in green is that when the mice are treated with this T cell activator inhibitor, there is a complete restoration of endothelial function. Then from there, he proposed that T cells expressing HIV protein are the cause of endothelial dysfunction and hypertension. The next question that he wanted to address is, what are the mechanisms of this endothelial dysfunction? Thanks to our collaboration with a thoracic surgeon, Dr. Vijay Patel, we had access to small pieces of aorta from either seronegative individual and people with HIV. And as you can appreciate on this Western blot, with people with HIV, we have an increased expression of the NADPH oxidase NOX1, which produces reactive oxygen species and has been shown to impair endothelial function. And based on those observations in human tissue, we measure the expression of NOX1 in the aorta of our wild-type and TG26 mice. And as you can appreciate on those graphics, then the aorta from the TG26 mice have higher level of NOX1. They also have higher level of NOx organizer 1 and NOx activator 1, which are necessary for NOX1 to produce reactive oxygen species and then induce endothelial dysfunction. Taylor repeated the experiment in the vessel of the animals that have been submitted to bone marrow transplant. Then when he did that, he observed that the vessel from the animals receiving bone marrow from the TG26 mice also expressed an increase in NOX1, an increase in NOX01. However, NOx activator 1 was not increased. But interestingly, again, when he did the rescue experiment where he transferred bone marrow from wild-type mice into TG26 mice, he reduced the expression of both NOX1 and NOX01. Again, we were interested in the functional relevance of those observations. Then the way to test the functional relevance of those observations was to repeat concentration response curve to acetylcholine in the presence of a specific NOX1 inhibitor, the JKT771 compound. And as you can appreciate, when Taylor used this inhibitor in the vessel from the mice receiving bone marrow from TG26 in gray, it completely restored the endothelial function, confirming what we have observed in ex vivo, in the aorta, showing that they have an increase in NOX1. We observe that blocking NOX1 restore endothelial function. To further confirm those functional data, we took advantage of our access to NOX1 deficient mice. Then what we did, we transferred bone marrow from TG26 mice into NOX1 knockout mice. And as you can appreciate on this first graphic representing the systolic blood pressure in our animals, when we transfer bone marrow of TG26 mice into NOX1 knockout mice, we don't observe an elevation in pressure, indicating that NOX1 is important for the elevation in pressure associated with viral protein expression. And then when we look at the endothelial function of those NOX1 knockout mice receiving bone marrow from TG26 mice, we observe that those mice are protected from HIV viral protein induced endothelial dysfunction. Then from there, what we propose is that T-cell expressing HIV viral protein induced endothelial dysfunction by increasing NOX1 in the vasculature, and those high level of NOX1 and ROS production also contribute to hypertension. Then from there, I can conclude that HIV-associated hypertension involves T-cell derived HIV protein, which elevates blood pressure via increasing vascular NOX1 expression and impairing endothelial dysfunction. Although those preclinical investigation in mouse model do not directly address human health disparity, we believe that the knowledge gained from this study will help treat the most vulnerable population, and notably the African American and gay bisexual population affected with HIV. And to finish, I would like to acknowledge the people who did the work, and especially Taylor, who was instrumental in the conduction of this project, and of course, our funding agencies. And now we will take a question. Then we'll take the question from the audience if they have any. In fact, we have a first question. In fact, before we start with the question, in fact, I would have a question for Dr. Moreira Bouchard, because you are looking at the effect of exercise. But if we look at the male gay population, there is a portion of this population that is heavy oriented towards exercise. And I was wondering if this portion of the population that is very heavy turned towards working out has basically a better cardiovascular function, and the fact that they work out so much basically mitigates the effect of stress, anxiety, and depression.
That's a great question. You're definitely right. There is a proportion of the gay male population who excessively exercise to cope with body dysmorphia that is very, very prevalent in the gay male population. And so I'm not sure in particular how that population's cardiovascular health indices would look, mostly because the exercise isn't necessarily to reduce stress. It is to reduce their stress, but it's a response to stress, almost like a maladaptive coping behavior. And the degree to which they exercise, particularly anaerobically, may negatively impact their health as their stress kind of positive feedback loop cycles worse and worse. So I'm actually not sure, but it's a really interesting question.
In fact, I would also have a question for Dr. Kwabi-Eddowes, and was very interesting to see this difference in meditation pattern. Then the question that I would have is whether the cause is, do you think is a cause based on whether basically the education and the social economic status could be contributing to these differences in methylation, or what could be the origins of those differences in methylation pattern?
Yeah, and thank you for that question. Yeah, so that's another interesting question. So to answer your question, is indirectly due to education and social determinants. But at the mechanistic level, DNA methylation is actually influenced by diet, a lot of stresses, hormonal changes. So the whole process of methylation is actually the addition of a cytosine to addition of methyl group to a cytosine ring to form the methylation. And this is directly the enzymes that plays role, the DNA methyltransferases, they are all impacted by factors such as infection, diet, stresses. And so that's why I mentioned indirectly, of course, education ties in with employment and income. And these are some of the things that can affect the type of food you eat, the sort of lifestyle factors that you expose yourself to. So yes, indirectly, education and certain social attributes can influence DNA methylation. But directly, the mechanism is definitely influenced by exposures to diet, virus, stress, and other modifying risk factors. Yeah.
Thank you. If I can follow up with another question related to that. Have you ever compared African American to African and seeing if those differences in methylation and if the difference in the rate of prostate cancer are different based on basically the origin of the people?
Yeah, so that's an interesting question. And there are ongoing studies where people are collecting prostate tissues from individuals in the African diaspora. And of course, this is one of the things that I want to do is to obviously compare the methylation level with those of the African American in the US. So again, you would think, I mean, without even necessarily doing the studies, you can project that there's going to be differences. Yeah, because we are obviously exposed to diverse environmental factors. So some of these differences obviously will be differences in diet, differences in lifestyle. And these are some of the factors that are obviously going to influence the differences in the methylation pattern. And also don't forget, a lot of this methylation pattern is actually transgenerational. So individuals from Africa who obviously have totally different set of environmental exposures would present differences in methylation pattern compared to their European counterpart. Yeah.
Yes, thank you. I think we'll take the questions that are in the Q&A, and there is a first question for Dr. Moira Bouchard, and the question is, might mechanism be different in subgroup of the queer community at a minimum based on biological sex?
That's a great question. Yeah, even the epidemiological data that we have from retrospective studies shows that there's probably an interaction of sex and sexual orientation. So this is interesting, right? Because it's not just sex as a biological variable, it's sex and gender identity and sexual orientation and probably race and socioeconomic status as kind of a really complicated set of social determinants of health that interact and are quite possibly unique in every single person based on the unique intersection of their identities. So lots of people, in my mind, are going to need to be recruited to large cohort studies such that we'll be able to slice and dice, the subgroups out with sufficient numbers to perform statistical analyses. Power is something I imagine will be challenging in building these studies because there are so many intersectional effects.
Well, thank you, Jesse. In fact, there is another question. The question is, Dr. Moira Bouchard, I'm curious if you have looked into health disparities between urban and rural people who identify as LGBTQA+ community. And the person say, I live in rural state and I am concerned about the folks in the LGBTQ in rural communities.
Yeah, that's something we haven't asked ourselves yet, just kind of getting off the ground. But I've thought about it in terms of, I'm sure there are geopolitical differences state to state, and then within each state, rural versus urban differences. And I suspect that they relate to two major variables. The first being structural inequity. So whether or not a given locality has laws or ordinances that prevent equal kind of opportunity and access and treatment. And then additionally, just basics of like healthcare access, right? In rural locations in the US, for any people, how far does the average person have to drive to go to the doctor's office? Where's the most local level 1 trauma center if there's an emergency? So healthcare access in rural areas is already, I think, well documented, but I suspect there's an additional effect when you combine it with structural determinants.
There's another question still for you, Jesse. Your talk has been very popular. Is there an additive effect on health disparities for racial ethnic minorities who are members of the LGBTQIA+ community? It's kind of related to the same question.
Yeah, similar question. I think so. Basically, the idea of intersectionality posits that every single layer of a marginalized identity you add on is going to contribute to a cumulative allostatic load. I would suspect that intersectionality is the most important thing to make sure we're comprehensively assessing when we're profiling risk for a given subpopulation.
Yeah, another question for you, probably for you, I would say, what pharmacological and lifestyle intervention would you propose for hypertension in young marginalized individuals and why?
That is the golden question. We still have to physiologically explore the mechanisms of this hypertension, this psychosocial stress-driven hypertension. There are less than a handful of papers that I'm aware of that have begun to do so. And so my answer is related to those papers which have demonstrated, for example, elevated diurnal salivary cortisol levels. So as we know, right, stress pathways, physiological stress pathways and psychological stress pathways converge on the HPA, the hypothalamic pituitary axis. And so I suspect there's a heavy cortisol component to the hypertension that's not dissimilar from Cushing's syndrome, which we think of as a secondary hypertension, but which is most often iatrogenic. And so I suspect in this subgroup of folks, they have more of what's really a secondary hypertension as it's driven from external stressors driving cortisol upregulation. And so knowing the way cortisol impacts the vasculature, I would say in contrast to the general scheme of ACE inhibitors and diuretics, these people probably need calcium channel blockers, direct vasodilators, since their vasomotor relaxative mechanisms are probably impaired.
Thank you. My next question probably for you, Dr. Quarbiado. What is something we as students can do to help decrease the impact of health disparities in marginalized community?
Oh, thank you. Thank you for that question. So I think, first of all, students need to be knowledgeable about the factors that is driving health disparities. So neighborhood communities, I think, is a big factor. So being at Howard, of course, we are located in a Northeast in a relatively minority neighborhood, which is relatively poor. So we are obviously more conscious about the health outcomes of individuals living in this area. So it's important for students to be knowledgeable about some of these factors. And neighborhood stresses, this is a big factor that is driving health disparities. Of course, diet is a predominant factor. Of course, poor diet is significantly associated with increased risk of some of these chronic diseases. Exercise, as we have talked about throughout this webinar, is a big factor. So it's for students to be knowledgeable about some of these factors. Again, education ties in with income and employment. And it's not necessarily the duty of students to really encourage other kids to go to school, but we know that one way to ameliorate a lot of these disparities is obviously through education. So if students have the opportunity to really advocate for individuals in minority population to, go to school, get education, those are some of the things that are going to bridge the disparities that we see.
Thank you. And maybe I think a professor needs some more training also to basically
exactly, especially the medical profession.
Students. Yes, because I think that will be crucial. I do not see more question in the Q&As, and I don't know if Dr. Kwabo or Dr. Mora Bouchard, if you have any other question. Because if we have no more questions, then I would like to thank our speaker for providing us with insight into our research and their significance to science and public health. And I also want to thank the audience for attending today and for the excellent question. So, and before you sign off, we hope that you will take a few minutes to complete a short survey about this session and your interest in the future webinar. Then thank you all, and also thank you thank you to the American Physiological Society for putting this webinar together.
Thank you for the invitation.
Thank you very much.